go, starting off another lecture for today. And so this is going to be over chapter 5. It's a survey of our eukaryotic cells and microorganisms. So these PowerPoints are available to you, so feel free to take notes and do whatever you need to, and let's get started. So this is over our Tolero's book, Foundations in Microbiome, right? And so we're going to go ahead and delve in. So let's start off with the history of the eukaryotes, right? So last video, we talked about how they first appeared at about 1.8 billion years ago. Over here it says 2 billion years ago. Overall, right, how they came about is that evidence suggests, right, that they evolved from a prokaryotic organism uh, having a symbiosis with another prokaryotic organism. And so organelles originate from this process, and how did that happen? Well, let's go ahead and look at that. So this idea that eukaryotic cells arose from this is called the endosymbiotic theory, right? And so basically, this just means, right, that eukaryotic cells, they came about after a very large prokaryotic cell engulfed a smaller prokaryotic cell. And somehow, this smaller prokaryotic cell survived digestion from the larger prokaryotic cell, and it's able to live within this environment inside this other cell. And we have a mutualistic relationship occur. They have a symbiosis, a mutual one, where basically the smaller cell, right, it receives protection uh, via the larger cell, right? It's inside that larger cell, it's protected from its environment, right? And it has ideal conditions to thrive. And so, how does the larger cell thrive? Well, this smaller cell is able to carry out specialized functions that the larger cell would not have done previously or would have had to work harder to do on its own. And so this relationship, it thrives. And so this cell that engulfed this smaller cell, right? It allows for it to have variability within its environment, and it also allows it to have a better ability to survive in its environment because it's able to do more because this smaller prokaryote within it is doing these specialized functions. And so eventually over time, right, this symbiosis, it basically evolves into a single entity. So I went ahead and made this image a little larger for you than what you have in your PowerPoints, but this basically kind of depicts what everything, what all goes on in this process, right? And so this larger prokaryote was most likely an archaean, right? And it probably exhibited a little bit more advancements than others. So you can see that they state that it probably had these little extra envelopes within the inside, right, of these internal membranes. But anyways, we get on, and this smaller prokaryote, right, we believe it's most closely related to rickettsias, right? Some very, very basic little cells, really interesting. We'll get more into those later, but they get absorbed by this larger cell, right? They get phagocytized, and it just stays within this larger this larger prokaryote, and it's able to basically metabolize and increase energy that's available to the host, right? And it does this aerobically. And we believe that this is how we came about to have the mitochondria. We believe that the mitochondria came from this smaller prokaryote. And later, right, in plants, we have the chloroplasts develop, right? So um, a cell that has mitochondria already, right? Already a eukaryotic cell, it has um, it engulfs, right, what we think to be most likely ancient cyanobacteria. It phagocytizes it. And basically the same processes happen over and over again, and so we get chloroplasts from this. So this is how we believe that many different protozoans and animals branch off. And we believe that, after the mitochondria, right, we develop algae and higher plants. And so, how do we know this? Right? Why do we support this theory so much? Well, we'll look at the two organelles that, call for this. And so, mitochondria, they exhibit a double membrane. And why is that important? Why would that be evidence? Well, if you look, right, it has its own membrane already. And when it gets engulfed by this other larger prokaryote, right? It continues to have that little membrane around it. And so from that we're able to kind of see this process, right? Um, Also, mitochondria have their own DNA. And not only do they have their own DNA, but their own DNA is incredibly similar to that of, you know, early early prokaryotes, right? They don't have a nucleus yet. So it's just this circular little plastid in there. And, you know, it's there, and mitochondria is able to do things that it needs to do. It's almost like its own little body within our cells. They also have their own ribosomes, right? And so the earliest of prokaryotes, right, even prokaryotes to this day, they may not have any particular organelles to build anything, but they do have ribosomes. And so for the mitochondria to have its own ribosomes really is a telling story. And so another good um, place for evidence, right, is that all eukaryotic organisms have a mitochondria or have mitochondria, multiple mitochondria, right? We all have this. So it just goes to show that without them, right? Without this single event occurring, we wouldn't have been able to have the rest of the eukaryotes, right? And so then later on, we have this happen with the chloroplast, right? To a particular um, uh, individual, right? That engulfs probably an ancient cyanobacteria. And we also see this double membrane, right? And we also see that chloroplasts have their own DNA, and it's circular, it's a plastid. And so with these, we're able to basically support the idea that eukaryotic cells arose from engulfing other cells and allowing for them to advance and further their functions and their specialties. Uh, Fun little fact, you can actually, uh, I guess, assign and look for different species, right? If you have an unknown organism, right, and it's a plant, you can actually run the DNA on the chloroplast, and they have particular genes that are highly conserved and just don't change among different individuals, and so you're able to uh, determine which ones are which. So that was like my thesis study. I worked on red algae, and so I did a barcode DNA analysis of a chloroplast plastid gene, and then I did a mitochondrial plastid gene, and I was able to determine what exact species it was. So yeah, mitochondria and chloroplasts have their own DNA, and it just further supports this idea of having endosymbiosis occur. So, what eukaryotic microbes do we have today? So, we have our protozoans, right? Our fungi and algae, and then our helminths. So, overall, right, we're going to be taking different looks at these different organisms. And so, in all of these organisms, right, we're going to look at this first top half, and this explains what all of these will have in their cells, or unicellular. So, all of them will have a lysosome. You know, they all need a little, garbage disposal, if you will. They'll all have a Golgi apparatus. All will have mitochondria. We'll also have filaments throughout the cell, microtubules throughout the cell. We'll have actin filaments, the cell membrane, right? And we are eukaryotes, right? So true nucleus. So we'll understand the parts of the nucleus. So the nuclear membrane, it's got pores, right, for transportation in and out of the nucleus. Um, the nucleus itself, and then we have the nucleolus, where all of the genetic information is. And then we have our rough endoplasmic reticulum. That one has the ribosomes on it. And then we have our smooth endoplasmic reticulum, which does not. And then in some cells, right, some of these eukaryotic cells, they'll exhibit a flagellum, a chloroplast. These are only in our autotrophs, right? Centrioles appear at a particular time, so it's not always in the cell. Then we have our cell wall in some organisms and glycolax in some organisms. So let's talk more about these individual organelles. Okay. And so to basically organize this, we're going to talk about the different structures within a eukaryotic cell. So they have external structures, right? Cell boundaries, and then organelles, right? And so these external structures can be things like glycolax, right? And in prokaryotes, they're like a capsule, but they can also produce slime. They basically help adhere an organism to the substrate of wherever it wants to attach. We'll get more into that. And then we're going to go over the boundaries of the cell, which would include the cell wall and then the cell membrane. We'll be going over, right, our cytoplasm, our nucleus, the organelles, different organelles that are used in locomotion, right, ribosomes, and the cytoskeleton. And then we'll go ahead and get into the big one, right, the nucleus. Some other organelles that we have is, endoplasmic reticulum, our Golgi complex, mitochondria, chloroplasts. Of those locomotive organelles, right? We've got flagella, we've got cilia, we've also got, within the cytoskeleton, we've got microtubules and microfilaments. And so, beginning with our locomotive appendages that are on the outside, right? We have flagella. And the flagella is just a long sheath cylinder. It contains microtubules in a 9+2 arrangement, right? And this is just important in that it allows for the for the flagella to function normally with a very rhythmic motion. It allows for them to slide past each other. And it's odd to the microtubule arrangement. You usually don't see odd numbers in nature, so it's just kind of interesting that 9 and 2 work so well. It's found in all flagella. So these flagella are also covered by basically an extension of the cell membrane. It covers it, so it's not just there, it is covered. It's part of the cell. And flagella for our eukaryotes are, of course, going to be thicker and larger to account for these larger cells. And so this is what we're talking about when we say the 9+2 arrangement. This is a cross section of, right, of our flagella. So here's our little pair in the middle, right? That's the 2. and then here are the 9 other microtubule arrangements. It's very odd, but it allows for it to move back and forth and twiddle around. Um, and that is basically the flagella, just locomotion. Cilia are basically like flagella, except that they're shorter, and when they're shorter and smaller, right, you're going to have more of them. They're going to be more abundant um, around the cell. And so they're only found in a single group of our protozoans, and only in certain animal cells. And so they function in motility, feeding, and filtering. So here is just an example of one of our ciliates right here. They do have a little oral groove, but yeah, we're looking at all the little small thin hairs around that allow it to move around. And so another external structure that we'd expect to see on some organisms within the protozoans would be glycocalyx, right? So the glycocalyx is an outermost boundary, and it is what comes into direct contact with the outside world. These are usually composed of sugars, our polysaccharides, and they are honestly a network of fibers that can, they can be a network of fibers. They can also just be something as simple as a slime layer or a capsule. And so basically, the glycocalyx just functions to adhere the little cell to different substrates, right? It also works as protection, and it can also work in signal reception, right? So that way it can understand the environment around it. And so underneath the glycolax, right, some fungi and most algae have a very thick, rigid cell wall. Okay, so algae and fungi have very thick cell walls. Protozoans, a few algae, and all animal cells lack a cell wall. So protozoans don't have a cell wall. Some algae don't have a cell wall, usually the single-celled ones. But yes, we don't have cell walls ourselves. We only have a membrane. What is the purpose of a cell wall? Well, it provides just basically structure and support, and it'll allow for the organism to have a shape. So fungi have a very thick inner layer of polysaccharide fibers, and those are composed of chitin or cellulose, and then a very thin layer of mixed glycans. Okay, and so algae, it varies in chemical composition. Um, what you can typically find in algae would be cellulose, pectin, mannan, silicon dioxide, and calcium carbonate, right? Um, those really small single-celled ones can look very beautiful with that calcium carbonate under the scope. The algae I worked on, it had a horribly rigid cell wall. It was the worst to extract DNA because that lysing step of DNA extraction, you're trying to break open the cell to release DNA. Oh my goodness, they were too supportive of those cells, so lysing stage took a very long time. Okay. So now we get into the cell membrane, and all of the eukaryotic cells have a cell membrane. It's just honestly a a layer, right, of phospholipids and proteins. And so, phospholipids, right, it's a bilayer because we have 2 rows of these phospholipids lining each other to produce this cytoplasmic membrane. You can also have sterols within them, and those just help to basically allow for communication with the cell. It also is a selectively permeable barrier, and so that means that not everything's just going in and out of the cell, right? We don't have that. It is selective. So only certain things are allowed in and out of the cell. And so with our eukaryotic cells, right, they all have these membrane-bound organelles, and they basically account for their almost entirety of their volume, so 60 to 80%. So just a little question, right? You can just answer this from, from your home, but which part of the eukaryotic cell is responsible for contacting the outside environment and also signaling between cells? Is it the flagella? Is it the cell wall? Is it the glycocalyx? Or the cell membrane? Okay, well, here's the answer. It's the glycocalyx. We have these different chemicals and enzymes on the outside of it that allow for signaling to occur and for it to be able to figure out what is going on. And so we're going to go ahead and look further into other organelles and their structures. This is a helpful little table. Feel free to like screenshot this or whatever as you will. But we have our nucleus, our nucleolus, our endoplasmic reticulum, our Golgi apparatus, the lysosomes, our vacuoles, mitochondria, chloroplast, ribosomes, cytoskeleton. And so talking about the nucleus itself, right? It's a very compact spheroid organelle, and it's honestly the first thing you see when you're looking at a cell. It's very large, it takes up a lot of space, and so it has a nuclear envelope that's composed of basically two parallel membranes, right? And they're separated by a very narrow space, and one of them is perforated with pores, and so that allows for transportation in and out of the cell, and so out of the organelle. And all of the nucleus, right, they contain chromosomes. It's not just a plastid anymore. No, it's a chromosome. We don't have just a simple circular DNA floating about. We have a chromosome. We also have a nucleolus, and this is the very dark area that is there to synthesize RNA, rRNA, and then ribosome assembly. And so this is what you'd see under the scope. We have our nuclear envelope. You can see within that it has little pores that allow for transportation in and out. The nucleolus, where our rRNA and ribosomes are getting synthesized. We also have chromatin, right? And so chromatin is just the wound up DNA, very very dark, right? And then we have our endoplasmic reticulum out on the outside. So this is a diagram of that photo, just a little better explained. So onto the endoplasmic reticulum, we have two types. We have the rough ER and the smooth ER. So rough ER, it comes from an outer membrane along that nuclear envelope, and it eventually extends out into the cytoplasm. And it's called rough because it's covered in ribosomes. And so from the rough endoplasmic reticulum, we synthesize proteins. And so these will then eventually get packaged off into, not packaged, it'll get sent off to just the smooth endoplasmic reticulum, right? And it's going to send it there for transport, but it's honestly the first step into our secretory pathway, meant for secretion of important sterols and stuff like that. And so we have our smooth endoplasmic reticulum, and this is a closed tubular network. It does not have ribosomes, and so that's why we call it the smooth one, and it functions in nutrient processing, synthesis of, what they're told by the rRNA, and so storage of lipids as well. So here's kind of a diagram of what that looks like in finer detail. And so here is our rough endoplasmic reticulum, right? It comes from this membrane from the nucleus. And you can see that scattered throughout it, we have some little ribosomes. Okay. And over here, we have our smooth endoplasmic reticulum. It has nothing on it. It's a little more tubey in shape. And from this over here, we can get transport vesicles that come to it and basically tell it, hey, we need this, right? And so we'll go ahead and start building things. So, internal structures, right? We have our Golgi apparatus. And so, what is the Golgi apparatus? Well, it's a little organelle that basically modifies, it stores and packages proteins. And so, it can be noticed by having just a stack of flattened sacs all across it. And, those are called cisternae. Okay. And so, let's talk about how we look at this process of synthesis, right? We can call it the transport machine. But we start with the nucleus, right? And it's, the brain of our cell. And it'll signal to the rough endoplasmic reticulum, right? It'll send it ribosomes. And so, once it has the ribosomes, right, they're going to need to have little transport vesicles, right? So after it's been told what it needs to make, right, transports those proteins that they made to go into the Golgi. Okay, so we first get the rough endoplasmic reticulum receiving that message, right? And then proteins are built, and then it's transported out into the Golgi. And then, right, it's sent out to vesicles, little transport objects, honestly. And then it's secreted out. And so we have things called condensing vesicles, and so those transport the proteins in two organelles, or even into secretory vesicles, and that exports that outside of the cell. So it can be within the cell and outside of it. So here's just a little image of that right there. So onto this, we have lysosomes. What are those? They're small vesicles that contain different enzymes that come from the Golgi apparatus. And so basically these guys, we like to say they're little garbage disposal organelles. They'll basically digest food particles and waste that we don't need. They also are involved in protection against invading microbes, so they'll dispose of them. We also have vacuoles, right? And so these are membrane-bound sacs that contain particles that are going to be digested or excreted or stored. They're basically just like little backpacks inside of the cell. They're just there to store stuff. So then we also have phagosomes, and those are vacuoles that kind of merged with a lysosome, and so it's not only storage, but it's also breaking down things at the same time. So over here, we're going to get on to the mitochondria. And what does the mitochondria do? It helps to produce and store ATP, the necessary energy that we need to carry out functions within our cells and our body. And so the mitochondria, it looks like a bean. It's a very spherical organelle. It has an outer membrane and an inner membrane. And inside of it, it has little folds, which we call cristae. And so the cristae membranes hold enzymes and electron carriers um, of aerobic respiration. So that's where that occurs. And so they can divide on their own, kind of further supporting that endosymbiosis theory. They can make more of themselves if they need. And so DNA and prokaryotic ribosomes are also carried in the spaces around the cristae, and that's called the matrix. And so here's a little image of that, right? We've got our own little ribosomes, the matrix, cristae. We have an outer membrane and an inner membrane, and our little circular DNA. Okay, let's move on to chloroplasts. Chloroplasts, they convert sunlight into chemical energy for us, for plants to use through photosynthesis. So you find chloroplasts in algae and plant cells. That's where you'll find them. Their outer membrane covers inner membrane folded into little sacs, and we call those the thylakoids. And then thylakoids are stacked into these structures we call grana, and these carry pigments like chlorophyll A and B, and then even other ones like phycocyanins, phycoerythrins that give you red colors and red algae. They're also primary producers of organic nutrients for other organisms to use. So very, very important organelle. And so this is what we're looking at right here. These are the individual thylakoids. And then this stack right here is the granum. Okay. And then we're seeing that double membrane, their own little circular DNA strand. And look, their own ribosomes. So why are ribosomes important? Well, they're composed of rRNA and proteins, and they're scattered throughout the cytoplasm and within the rough endoplasmic reticulum. And so they're larger than prokaryotic ribosomes, but what it's important to know is that they function in the synthesis of proteins. All they need is that rRNA, and they can produce proteins. And that's the simplest and most necessary thing to have in a cell, aside from your genetic material. And you can find them just all throughout the cell. So let's move on to the cytoskeleton. And the cytoskeleton is basically a flexible framework of different proteins, and these can create microfilaments and microtubules. And they're just a network that's formed throughout the cytoplasm. They can be involved in anchoring an organelle to a specific spot. They can also help in the movement of cytoplasm. So, like amoeboid type motion, right? It's using these to kind of uh, get around. And so they also are able to be transport and structural support of the cell itself. And so we're about to go to this next table, which you are more than welcome to print out or screenshot, however you like, to help you study. But these are just some different kinds of like a key. So the key is that if it has a positive, right, it means that most or all of the group exhibits that characteristic. If it's a negative, it means that they most likely lack it. They're not going to exhibit that. And then if it's a positive minus, it means that some members have it, some don't. And so viruses, they cannot do their own metabolic or genetic activity at all. They rely on a host cell for that. And we're going to look at that table in a little bit. And prokaryotic types have very different structures. And so another side note that they'd like you to know is that very, very small and even larger bacteria exist. And so nucleic acids, of course, all cells are going to have that. A true nucleus, only eukaryotes have that. So that means they'll be the only ones to have a nuclear envelope. Prokaryotic cells are the only ones that will have a nucleoid to hold their genetic material. And then mitosis, that only occurs in eukaryotic cells. Production of sex cells, some prokaryotic cells do, and all eukaryotic cells do. Both can exhibit binary fission, right? And then, for biosynthesis, right, like they can both independently create what they need, right? However, eukaryotic cells are the only ones to have a Golgi apparatus or the endoplasmic reticulum. However, within these, we do have ribosomes in prokaryotic cells and eukaryotic cells, as discussed earlier. For respiration, right? Both prokaryotic and eukaryotic cells use enzymes. However, mitochondria are only present in eukaryotic cells. Photosynthesis can occur with pigments, and those can be present in prokaryotic cells, and they can be present in eukaryotic cells as well. They're only in eukaryotic cells if they have the chloroplast, and so chloroplasts only occur in eukaryotic cells. For motility, right, we can have flagella in both prokaryotic and eukaryotic cells. Cilia are only going to be present in eukaryotic cells. Cell membranes are going to be present in both. Cell walls are going to be present in both. Viruses do not have a cell wall, instead they have a capsid. And then we have the capsule, which can occur in eukaryotic cells and prokaryotic cells. And so this kind of shows you the difference in sizes. Prokaryotic cells are always going to be much, much smaller than eukaryotic cells because eukaryotic cells have large organelles and a large nucleus. And then viruses are going to be much smaller than the rest of these cells. Incredibly small. So let's go ahead and look at the phylogeny, right? The relationship of these eukaryotics to one another. And so we have our red algae, which we call archaeoplastids. Those developed from a very early ancestor that was probably an archaean, right? We have our SARS, right? And these can range from all types of different organisms, from brown algae and diatoms to apicomplexans, dinoflagellates, ciliates, and then our rhizarians. I love foraminiferae. They're so beautiful to look under the scope, and so are radiolarians. If you ever get a chance to look at them, oh, just beautiful. And so these kind of diverge off from a trait right here, where we get these two different groups, and that would be the forams versus our diatoms. And that is honestly, I believe, just what causes that branch is a difference in the chemical makeup of their tests. And tests are just basically like the skeleton of these very, very, very small organisms. So one is carbonate, and then the other one is silica. And so then we have our dinoflagellates, right, who also share that same characteristics. And then we have our ciliates. And then over here we have our Escavates, right? And so we have our diplomonads and our euglenoids, our kinetoplastids, and our parabacillids, right? And so right here they had a common trait that just kind of branched them off into separate groups right there. That was their last common ancestor there. And then we have us, right, over here. And so we came from this group called the Opisthoconchs, right? And so that separates the fungi and animals from the amoeboids. And so out of all of these organisms, we're most closely related to the fungi. But yeah, these little nodes right here show the last common ancestor that these organisms had in relation to another. Oh, that's said right here. And so we're going to look further into these different microorganisms, and let's get started. First, let's get into fungi. And so a majority of these, they can be unicellular or colonial, but a few have some cellular specialization within that. This is a very large group. We have over 100,000 species, and as we had discussed in lab, we can divide these into uh, two groups, right? Um, well, we had discussed these two groups right here because we were looking under the microscope. But you can divide them into these two groups, which would be the large ones that you can see, macroscopic, right? And those are our mushrooms, our puffballs, our gill fungi, our toadstools, things like that. And then we have our microscopic fungi. And so those have two different morphologies that we can further divide them into, and that would be the hyphae, which are long filamentous fungi, right? And that's found in our molds. And then we have yeasts, right? And these are round ovoid shape, and they only have asexual reproduction. And so with fungal hyphae, right? These are very long thread-like cells, and they make up most of the body of these filamentous fungi, right? These molds. And so they present various textures of mycelia, right? And so mycelia is when you have a whole lot of these filamentous threads, right? And so you can also get an array of different colors just due to the difference in spores. And so, yeah, right here, individually, those would be little hyphae, but this whole mat we're going to call the mycelia, right? And some can have uh, septums, right? So, like little divisions within themselves. And so when we get onto yeasts, right, these are distinguished by having a very round ovular shape, and it only reproduces asexually. So yeasts, you know, they'll bud off of off of each other, and they're gonna just maintain that way. And so it presents a cell wall, but it lacks locomotor organelles within it. And so they aren't able to move themselves. Um, they can only have these little buds that can eventually be pushed around throughout the world. So here's some more examples of budding. All fungi are heterotrophic. Most though are harmless. They live off of dead plants and animals. But some can be parasitic and they can live on our tissues. They can live on other organisms. However, none are obligate parasites, meaning they don't have to parasitize as a means of survival. It's an option. And so these parasites can cause what are called mycoses, and mycoses is plural for fungal infections. If you have a mycosis, that's an infection caused by a fungus. And so these can be incredibly widespread. They're found in all types of habitats. So right here we have our little citrus fruit, right? and we have some mold growing out on top of it, and that's its little fruiting body right there under the scope. This is attacking, well, not attacking, but it's feeding on this outer layer of your skin. And right here we can see the little hyphae, right? And it's septate, meaning it has these little divisions within itself. And so our molds, right, which under the scope are very filamentous, when you see them with only your bare eyes, they're going to appear to be cottony or hairy or kind of velvety. And that's just because they have all of that intertwining hyphae, right? That mycelium. And so that's what we're seeing that produces that texture. And so we do have vegetative hyphae that are used to just digest and absorb nutrients, and then we have reproductive hyphae which help to produce spores in the reproduction of these. We also have yeasts, right? And so some have pseudohyphae, and that would just be a chain of these little yeasts forming when there's multiple buds just attached in a row. But they don't have hyphae. It's almost like a hyphae. And because of that, it's not considered a true hyphae that we would see in molds. And so this is just an example of that, where they're all just stacked upon each other one by one. And this is a true hyphae right there. And so, as we had said, right, they mostly reproduce through spores that are formed at the ends of our hyphae, right? We have that sporangium form, and within that sporangium, we have sporangiospores. And the sporangiospores will eventually become a spore that will eventually grow out a hyphae as it germinates, and it can continue that cycle. Sporangiospores, right, they're formed by a bunch of cleaving within its little sac-like head that's called the sporangium. And so spores are initially enclosed within the sporangium. However, once the sporangium bursts, they're just completely released. So then we also have these things called conidiospores, right? And so these are free spores that just they don't have that enclosure of the sac around it, and so that's how those are different. So here we have an example of a sporangiospore, right? It's enclosed in this little sac right here. And these two, if you look very closely under the scope, these would be, surrounded by a little membrane layer. But over here we have conidia, right? And so these are, they're not covered by a membrane. They're just free floating out there until they are released. And so how do spores come about? Well, they're formed how are they formed in the sexual sense? So spores can be formed following the fusion of two different strains, and they form a sexual structure. So an example of that would be with our zygospores, our ascospores, and our basidiospores. And we can use these spores to classify these different organisms. And so we're going to go ahead and look at this mold on the bread, right? And it'll two sexually reproduce, right? We'll have these two different strains, right? This positive and a negative. And so they'll contact each other, right? And it'll be like a mating kind of situation there that'll eventually lead to fertilization, and that allows for a zygospore to form, and eventually it'll mature, and from that we can get this little uh, zygospore producing sporangia, and we can get the spores from that. And so within this kingdom Eumycota, right, it can be divided into several different phyla based upon just sexual reproduction. And so within the zygomycetes, right, they're in phylum Zygomycota, and they produce zygospores. And so you can see sporangiospores and sometimes conidia. Um, Within the Ascomycota, or the sac fungi, ascospores are what they produce, and they can also have conidia. In the basidiocarps, right, in the Basidiomycota, they produce basidiospores, still producing that conidia. And then we have the Chytridomycota, sorry for messing that up, the Chytridomycota. And these are just flagellated spores. And we have fungi that also only produce asexual spores. And so these are different ways you can classify the fungi. And so you're also able to isolate them using different media. And so there you can observe macroscopic and microscopic of these different asexual spores. You can look at hyphal types. You can identify them based on their colony texture and the color that arises from it, other physiological characteristics, and you can even, do a DNA analysis and see how you can what specimen you have, and that's what you can do to identify these. And so we have different representatives of these groups, right? And so in the phylum Zygomycota, right, they have the sexual spores that we call zygospores, and then they also have asexual spores, and those would be sporangiospores, right? They have that little sac around them. Sometimes they have conidia. So they're hyphae, right? In the Zygomycota, they will not be septate, meaning it will just be one complete hyphae. But if they do have septa, they're gonna be complete and distinct. However, most of these species, they're free living saprobes. Only some are animal parasites. Uh, they can be unfortunate contaminants that are kind of just annoying to have in the laboratory, whether it be from food spoilage, and because of that, they can also be very destructive to crops just by contaminating them. And so we have some common molds, right? The Rhizopus, which is the black bread mold. And then we have like Mucor, Absidia, Circinella. And so, we can just continue to look at these. And so this is a common um, fungi that appears in soil and in plants. This one is known for having a little curve right there. It looks almost like a little wilted dandelion. This is Circinella. We also have some from the genus Histoplasma, and it causes Ohio Valley fever. We also have the Trichophyton, and this is one of the causes of ringworm. And ringworm is just a common name for having this kind of infection that grows in a ring-like pattern, but they're not always related. They just typically grow like that, and so it's just a common name. We also have those in Coccidioides immitis, and so this causes valley fever. Then we have Candida albicans, and so this is a cause of different yeast infections. And then we have Staphybotrys, and so that's a toxic mold. Okay, and we looked at this one in lab, the Aspergillus. And this is a really nice scanning electron microscope picture called a micrograph of a Candida pseudohyphae species. And so we have sexual reproduction that occurs by basidia and basidia spores, right? And so this is going to be with the uh, basidia group, right? And so they have asexual spores. Those would be called conidia. And then they can have incomplete septate hyphae, right? And so they're not going to be always fully septate or fully separated in each of those. And so they'll have very it's common to have fleshy fruity bodies with this group. And so an example of that would be like a large mushroom. We have our puffballs and our bracket fungi. And then we also have some plant pathogens that you can see, and they're very squishy. That's what it means by like fleshy. And so we also have one human pathogen, and that's in the yeast. That's called Cryptococcus neoformans, and this causes basically an infection within the body in several different organs, which can include the skin, the brain, and the lungs.